जी आहे ना, आपण ती लाईव्ह लिंक सेंड करतोय.
आताच
हा
रेकॉर्डिंग नाही सुरू होणार.
रेकॉर्डिंग मी चालू होणार ती दिसायला दिसेल तुम्हाला, पण आपल्याला अजून ती रिमूव्ह करायची जी आहे ना ती आपण अजून अपलोड नाही केली. हा फक्त आपल्याला सध्या दिसते.
सुरुवातीला ती गोंधळ घातला.
नाही ते आपण अपलोड नाही केली ते अजून. ते फक्त आपण लाईव्ह दिसतंय ते आपण रिमूव्ह करणार आहे आणि अपलोड करणार आहे. त्याच्यामध्ये आवाज वगैरे हे करून परत अपलोड करणार आहे. आता हे दिसणार आहे आता आपण
मराठीचा फारच गोंधळ झाला. कोणाला टाकायची
हा हॅलो मॅडम, आवाज येतोय का? चेक करा ना. मॅडम येतो का आवाज? चौधरी सर आवाज येतो ना क्लिअर? काही बंद इथं मी काय करतो का, इथं काय म्हणतो नाव ऍड करा. माझा आवाज येतो मेसेज
मेसेज टाकू ना
अरे हा
आता पण
हो ते आवाज ऑफ
Hello, Peter sir, can you hear me? Hello, Peter sir. Yeah, hello. Bandwidth is low. So we can't hear your voice. Just please remove your mask. Because he's having a lot of problems. Hello, sir. Can you hear me? Hello? Hello, so your audio is off. Please unmute.
Okay, now?
Yeah, sir.
Hello?
Yeah, sir, we can hear you now.
Okay, you got it. Okay, this is the
But I'm
first
not
time
able
I'm doing
to
this.
see you. So you're having a very low bandwidth.
I'm in a rickshaw at the moment. I'm in a rickshaw at the moment. So,
Yeah, okay. So,
anyway,
hello, sir.
yes, tell me now.
Okay, so you'll be going to some place now, so you'll be stable over there.
Yes. At the moment, I'm on the road. This is a test call, isn't it?
Yes, sir. Huh? Okay, so when you reach at your place,
Yeah, I figured out. So you're,
just tell me. That time we will test it again. Okay?
pardon me?
कॅमेरा इकडे धरा सेंटरला सरळ 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 ढकल तिकडच ढकल नाही पलीकडं 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 ढकल ढकल हा बस हॅलो 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 बाकीचं बंद कर लाईट्स बाकीचे लाईट्स बंद करा हॅलो चेक हो येतोय
हॅलो माझा येते ना आवाज
हा त्यांचा नाही येत आहे
हॅलो येतोय आता
थांबा एक 2 मिनिट
आवाज येतोय
एवढा मोठा आवाज नका ठेवू
हॅलो
एवढा आवाज मोठा ठेवू नका आवाज एकदम जोरात वाढेल तो हा करा
ऑडिओ ऑन आहे हॅलो आवाज येतोय आता सर इथल्याच ह्याला आवाज येतोय हा
हॅलो चेक
येस सर तुमचा आवाज येतोय तुमचा आवाज येतोय सर हॅलो
स्पीकरचं चेक करा तेव्हा आता थोडासा आवाज
हॅलो येतोय आवाज
हो झाला चालू
ओके
आवाज कमी करा
येतो ना सर आवाज
जरा पुन्हा एकदा टेस्ट करा
हॅलो कॅन यू हिअर मी
नाही
नो
हॅलो
हॅलो सर या सर हॅलो 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 सर कॅन यू हिअर मी हॅलो सर
Oh, hi. Hello.
हॅलो हॅलो येस सर
Hello.
पीटर सर
Hi. Hi. Say goodbye. Now I have to test. Yes.
पीटर
I
सर
have
योर
to get a
वॉइस इज
COVID
ब्रेक
test done.
ओके
I have to get a COVID test done. I'll see you in 5 minutes.
येस सर ओके
Test करना था. के नीचे but yeah. Okay, thank you.
नाही ही बरोबर आहे. फक्त स्पीकर साठी तुम्ही ती पिन काढून आणि पुन्हा एकदा त्याच्यात दुसरा स्पीकर आण तिथून कुठून दुसरे स्पीकर आण कुठून नाही तर साऊंड टेस्ट असं नाही ना वरती उभं राहून बघा ना थोडं वरती
हॅलो, आवाज येतोय सर? हॅलो हॅलो, कॅन यू हिअर मी सर?
नाही नाही तो कुठला स्पीकर तो नाही तो आहे का नक्की नाही हा डेल स्पीकर आउट तो नाही ना आत 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 करून ठेवलं तरी चालेल
हॅलो, आवाज येतो का?
हा, माझा आवाज येतो क्लिअर?
हॅलो, ओके राऊत सर.
This I'm learning. I'm learning first.
हॅलो राऊत सर,
हा, येते का आवाज?
येतोय आवाज, बारीक येतो.
क्लिअर आवाज येतोय का माझा?
हा येतोय सर, थोडं कमी येतोय.
आवाज थांबत येत नाही माझा.
हॅलो,
तर पिन टाकायला.
डेल स्पीकर.
हं?
डेल स्पीकर.
डेल का?
हॅलो सर, कॅन यू हिअर मी? हॅलो,
तिकडे सुरू केलंय तुम्ही? हं?
हॅलो सर, कॅन यू हिअर मी नाउ?
नाही नाही.
स्पीकर
बदला बरं. हं. हं.
शिंदे सर.
tomorrow have to be there.
हॅलो मॅडम. हॅलो हॅलो मॅडम.
हॅलो हॅलो. हा सर. हा राऊत
येतो का
सर.
आवाज क्लिअर माझा?
आवाज बारीक येतो सर.
आवाज बारीक येते का? क्लिअर आवाज येत नाही का?
तुम्ही समोर या म्हणजे आवाज येतोय थोडासा. हॅलो.
हा आवाज क्लिअर येतो का एकदम?
हा येतोय
हो हो.
बट आवाज व्हॉल्यूम वाढवा. हॅलो हॅलो.
हॅलो हॅलो.
हॅलो सर. हॅलो सर. राऊत सर.
म्यूट करायचं ना ग.
कोणाला मगाशीच केलं ना. Hello, Raut sir, can you hear me? Hello.
Yes, ma'am.
Yeah. Very soon we will start with the webinar. So please, all the participants are requested to mute yourself so that there will be no disturbance in the middle of webinar. Don't present any kind of screen. Please take care that you have muted yourself.
Your video is stopped. Your mic is unmuted. Unmute the line.
Hello, check.
Hello, sir. Yeah.
Huh?
Principal, sir, we
I'm
can hear
getting.
you.
Yes. Can you hear me?
Yes, sir, I can hear you.
Hello. Yeah.
Hello,
Okay.
sir.
Yeah.
Yeah, we can hear you, sir. So,
Yeah. Good.
okay. Should we start, sir, with your permission?
Yeah.
Yes, sir. Thank you. Okay, so am I audible to everyone?
Yeah.
A very good morning to one and all. On behalf of Ray Shikshan Sanstha's Rao Bahadur Narayan Rao Borauke College, Sri Rampur, I extend my warm welcome to all the students, research scholars, faculties, and all the participants from different states of this nation who have actively participated in this webinar organized by Department of Zoology and IQSC. So I give a warm welcome to all the participants over here. So it's a pleasure to have you all on this occasion, because if you see in this pandemic, there are bunches and series of webinars all over the world. And Department of Zoology has taken one step to give us some knowledge on the topic contemporary trends in zoology. Because as we see, as the decade goes, there are always some trends in lifestyle or different kind of things. So things has been changing, evolving along the decades. But as a subject knowledge, the things are also evolving, the trends are changing. So we have taken one step that to know what are the current trends in our subject, that is the zoology. So here we have called upon the two resource persons, our guest of honor, Mr. Peter Smerachek from Uttarakhand and Dr. Meghna Joshi from Department of Stem Cells and Regenerative Medicine from Kolhapur, D.Y. Patil Education Society, Kolhapur. So we have great eminent personalities today on this national webinar. So hope you all will enjoy this both sessions. So to begin with the webinar sessions, I request our convener and head of the Department of Zoology, Mr. B.N. Kaunti sir, to introduce and explain the objectives of the webinar. Over to you, sir.
Hello. Good morning, everybody.
Good morning.
On the occasion of today's webinar organized by Department of Zoology and IQSC, RBNB, Sri Rampur, I welcome to all on the dais. Honorable Minatai Jagdhane, Member Managing Council, Rayat Shikshan Sanstha, Satar. Our respected principal, Dr. N.S. Gaikwad, Dr. P.V. Bade, IQSC coordinator, respected resource person, Peter sir, Dr. M.G. Joshi, my colleagues, and dear participants. The purpose of seminar entitled Contemporary Trends in Zoology is to create awareness among teachers, students, and society also. There are two resource persons and their topics are first, recent trends in classification, taxonomy, and another one, stem cells and stem cell therapy. It is related to medicine. It is related to medicine. The first taxonomy, it is mainly concerned with butterflies, insects. In India, there are about 1,400 species of butterflies. And butterflies plays important role. They are ecological indicators or biological indicators of urbanization because they are very sensitive to microclimatic change like temperature. They help in pollination of crops and other wild plants also. About more than 50 different crops, vegetables, fruit plants are fertilized by the pollination caused by different butterflies. Besides pollination, they also help in recycling of nutrients like NPK. And next one topic is recent one, stem cells and regenerative medicine. The stem cells are in multicellular organisms undifferentiated cells or partially differentiated cells. They are transformed into a particular type of cell, like muscle cell, nervous cell, blood cell, intestinal cell. And these stem cells are used in medicine. The scientists and doctors are interested in the use of such stem cells in different diseases to prevent disease or to cure disease. Generally, they are used. Stem cells are produced initially in bone marrow cells. They are then again found in brain cells, blood vessels, skin, etc. And these cells are useful in controlling various diseases, that is prevention or treatment of disease. Generally, spinal cord injury, type 1 diabetes, then Parkinson's disease, burn, heart attack, strokes, etc. Hence, this one is recently important branch of medicine, use of stem cells in different diseases. Thank you very much for your kind attention. And I again welcome to all participants to enjoy these important sessions.
Thank you, sir. Now I request respected principal of our college, Dr. N.S. Gaikwad, sir, to deliver presidential address on this occasion. Please, sir.
Honorable keynote speakers, Mr. Peter Smetchek, who is founder of Butterfly Research Institute, Bhimtal. He is also member of State Wildlife Board, Uttarakhand. The another keynote speaker, Dr. Meghnath G. Joshi. He is heading from the Department of Stem Cells and Regenerative Medicine from D.Y. Patil Education Society, Kolhapur. He is also managing director of STEM Plus Biotech Private Limited, Sangli. Honorable Minatai Jagdane, who is member of our Rayat Shikshan Sanstha, Satara. She is also heading as the chairman of Rao Bahadur Narayan Rao Borauke College, Rampur. Honorable head of the department who just introduced you about the seminar, Professor Kalunkhe, Mrs. Shinde, Dr. Bendre, all the colleagues from the zoology department, all the other participant colleagues as the teacher participants, all the student participants and their friends. I am very happy to be here to share my thoughts on the behalf of the college on this national webinar on contemporary trends in zoology. As the Rayat Shikshan Sanstha is concerned, it has crossed its centenary in last year. Now, the old prestigious ethically working institution, a group of institution is widely known in Maharashtra as Rayat, and it is working with its ethical practices in especially Western Maharashtra. This college, as far as the college is concerned, as it has crossed 60 years, and within this 60 years of span, it has changed its total structure, both way in quantitative as well as in qualitative. College is enjoying the A+ status from NAC with CGPA 3.44. College is also having its ISO status, and I'm very happy to share the views about the college, as it is the prestigious college in the Maharashtra, having very good CGPA score. As far as the department is concerned, as I congratulate all those organizers that they have chosen very good subject for this webinar as the contemporary trends in zoology. As far as zoology, we consider as the study of animals or animal kingdom. We study the structures, we study the classification, we study the embryology, we study the evolution, habits of animals, etc., etc. Being students of zoology, we all are interested in studying all these things. But what is the current scenario in zoology is very important. People are talking about the system, and from system to organisms. People are talking about the cells, people are talking about the tissues, and the animal tissue culture, ATC, is the most promising fields nowadays, and it's the very good application of zoology subject. As far as when we consider the uses of this animal tissue culture, we know that basically we consider the cell biology, spatial cell functions, we have cell cycle mechanism. This type of study is also including the toxicity testing for few drugs, studying cancer cells with respect to the role of chemicals, role of viruses, role of radiations of the cancer cell, production of vaccines, production of viruses to make the vaccines. In this pandemic situation, as we are worried about this COVID-19, so people are also trying to find out the vaccine for this COVID-19. So these all things, they are included in the study of the basic subject zoology. As far as the today's topics, as Professor Kalunke already told you about the Butterfly Research Center and Mr. Peter, who is heading as the founder from this Butterfly Research Center. And as far as we people are concerned, especially environmentalists, they look after these type of butterflies as the signaling, or they will be providing us good signals about what the climate is going to change. That's why the butterfly research is very important field, and Mr. Peter will flash good light over the topic. Another field taken by the keynote speaker, Dr. Meghnad Joshi, as Meghananda Joshi. So he's working on stem cells and regenerative medicine. So as Mr. Kalokhe has already told about the stem cells and its use. So there are many things to be discussed, and very good brainstorming will be for my teacher colleagues as well as students to learn much more things about the recent trends in zoology. With these views, I congratulate for choosing this very nice topic. I congratulate all the participants. I welcome all the participants for being with us with this webinar. And I hope that webinar will be, I hope that the webinar will be fruitful for all the teachers and students. With these words, I conclude, and I welcome Peter sir. I welcome Joshi sir for being with us. Thank you very much.
Thank you.
Namaste.
Thank you, sir.
Namaste.
Thank you, sir. Now to begin with our first session, I request Neha Bendre ma'am to introduce today's resource person. Bendre ma'am.
Thank you, Pomat. It's my great pleasure to introduce our today's guest of honor for the first, our speaker, Mr. Peter Smeracek, sir. An honor. It's a privilege that you accepted our invitation and said yes. Despite of you having a very dangerous range issues over there, but he just managed to come up with that range issue and accepted our invitation. So it's a really pleasure that you are here now. It's my privilege again to introduce our today's guest, Mr. Peter Smeracek. He has a very big biodata, but I'm going to make it very short. So he has completed his graduation in the University of Kamau of Nainital. Founder trustee of Butterfly Research Trust and founder of Butterfly Research Center and a former member of State Wildlife Advisory Board, Uttarakhand. He's a lepidopterist. He's a butterfly man. He loves butterflies. He loves to do research on butterflies, moths. And so he's a lepidopterist who has carried his research mostly on the butterflies and moths. Serves as a consultant and is a recognized determiner for Lepidoptera vide, the Directory of Determiners 2005. He had identified Lepidoptera at University Museum at Oxford, United Kingdom. So it's a great thing. Also at the National Museum of Natural History, New Delhi, the Forest Research Institute, Dehradun, Forest Research Institute at Tabo, Bhutan, Indian Institute of Sciences, Bangalore, Rainforest Research Institute, Zohar. He has donated on 2,000 plus identified duplicates of Lepidoptera at National Zoological Survey of India, Kolkata, and Dehradun. He has also invited to address to the University of Entomological Society at Jesus College, Oxford University, United Kingdom, about his work on Lepidoptera as bioindicators. Okay, so he was also a consultant of Limca Book of World Records for natural history for 10 years since 1997. He has completed various research projects funded by Times Research Fellowship, which has been granted by University of Oxford, United Kingdom. Rafford Small Grant projects also has been granted to him. Many research projects are there, so I will not name all of those. He has, most important thing in Nainital, he has a huge museum at his home. So definitely, whenever we get chance, we want to visit his home as it contains a lot of varieties of butterflies. And he is inspired, means he is collecting, but also he is donating at the same rate. So he has over a dozen of Lepidoptera and taxa belonging to 7 families described by him. It includes 1,700 moth species and over 300 butterfly species. So imagine what kind of house he must be having. So making it the 4th largest reference collection of Lepidoptera in India. So duplicate booklets have been donated by him at various national collections. Among many accomplishments, he is a prolific writer. He's the author of 6 books, same on the Lepidopteran. He's the editorial-in-chief for many of the national and international journals. Then he has 63 publications to his credit with 281 citations. And lots many more, but I will stop over here. He also has a newspaper column of his own, okay, on fortnightly column on ecology in the Times of India, in Times of India, and he has popular series, The Study of Resonance. Still, there are many, but I will leave all to you that as you begin with the session, you will get to know him better and better. So, Peter sir, thank you once again for accepting our invitation. And
Thank you for having me, Neha, and thank you for embarrassing me more than I have been embarrassed in the past, I don't know how many months. So,
Thank you, ma'am. Now I request Mr. Peter Smetajek, sir, to deliver lecture on current trends in taxonomy. Please, sir.
yes, okay, good. So, is that better?
Yeah.
Oh, fine, okay. Thank you. So, thank you very much for having me here. And let's get on with the topic. The topic that I chose was current trends in taxonomy. And we're talking about current trends in biology. So taxonomy is the root of biology. Is it okay to talk in Hindi or English?
In both languages, maybe.
Okay. I'll talk in English and see what happens.
Yeah.
So just 2 minutes about the history of taxonomy. So it started off with the need to name things. So all of us have names to distinguish us and to attribute our good deeds and bad deeds to us. So the same way when in ancient communities, plants, animals, whatever of interest to the community were given names. So you had rose and daffodils and chrysanthemums and whatnots, as well as bees and flies and butterflies, as well as bears and jackals and lions and tigers. And in due course, when communication improved, communities came together, and the need arose for universal names. Now, what happens with native names is that I live in the Himalaya, as Neha Madam has told you. And over here, we have leopards and tigers. In the plains, you have lions and leopards and tigers. Now, in the plains of India, a sher is a lion, a baag is a tiger, and a tendua is a leopard. When you come to the hills, we don't have lions. So here, the tiger becomes a sher, and the leopard becomes a baag, and nobody has heard of the tendua. So if a hillman tells you that he saw a baag in a tree, he means he saw a leopard in the tree. You will not believe him because tigers do not climb trees. So you'll say the hillman is telling lies. But actually, he's telling the truth. It's just that he has a different name for it. So to avoid confusion like this, it was decided that it would be good if there were universal names for things. So the system of naming things evolved, which is the root of biology. Right? Once we find out something about an organism or a plant, we want to hang all that information on that peg. We don't want it being confused with other things. We don't want the information that a rose has thorns, a rose bush has thorns. We don't want that confused with a wheat plant, so that people get the wrong idea that a wheat plant has thorns. So we give them different names and put all the information that we can gather on those particular names. So naming is very important. Now, names require rules. And most important, what is to be named? So the unit of biology is the species, right? Linnaeus, 200 years ago, Carl Linnaeus, a Swedish naturalist, he perfected the method of naming things. So he started what you all know as the binomial system, so that you have a genus and a species for each thing. And the problem was that he called everything a species, but he did not define what a species was. So Linnaeus did not know what a species was. And you will get a better understanding of what how Linnaeus saw the world, if you consider that Linnaeus was wondering where angels fit in his classification. So Linnaeus believed in angels, and till the end of his life, he didn't know where to put the angels in, right? 100 years passed, things began to get names, and the species was left undefined. 100 years later, Charles Darwin came up with the theory of evolution. And in that, he claimed that species were not fixed from before, but they evolved into each other. Okay, so a lion could in due course become a tiger if it lived in the jungle. So it didn't need the hair around its neck, and it needed stripes to camouflage itself. So it became a tiger. So now that was unacceptable to the establishment, because they were following the Bible, which said God created everything. And then Darwin came along and said, things created themselves. So that was absolutely unacceptable. So there was a huge thing against him. But even Darwin did not define a species. There's no definition for what a species is. The species was defined in 1942, 90 years after Darwin. In 1942, it was defined as if two individuals mate, if they can produce fertile offspring, they are the same species. If they cannot produce fertile offspring, they are different species. That is called the biological concept of species. So, for example, you have very many different types of dogs, but they can mate, and the babies they produce can also produce babies. So those two very different types of dogs are actually the same species. But you have a donkey and a horse, and they will mate and they will produce a mule, but the mule cannot produce children. So the donkey and the horse are different species, right? So that's the biological concept. It was followed for some years, and then of course the Americans came up with 20 more definitions for species. And overall, the other definitions did not work. So we are left with the biological concept of species. Now, how do you, ideally, in an ideal world, we would take all the species and crossbreed them and see if they produce hybrids. And if they don't produce hybrids, then we will say, okay, they are different species. If they do produce hybrids, we will breed those hybrids, and then we'll see if they get fertile. And then we will decide whether they are species or not. Now, you can't do all that. So we have to follow other things, other methods. Until 100 years ago, we used to follow physical differences. So if a dog and a gorilla look different, they're obviously very different species, right? So species were defined on the basis of the way they looked. When it came to insects, then often the look was not enough, because you have mimicry and all those sort of things. So the next thing became that 100 years ago, in the early 1900s, they started the study of, at least in insects, the bony structure, the chitinous parts of the male genitalia, because the bones were different. And there was a theory which they call the lock and key. So they assumed that all the male genitalia of a particular species has to look the same, because it has to fit a certain type of female genitalia. And therefore that was the lock, the female genitalia being the lock, and only a particular key could unlock it. So they followed the lock and key. When you get down to genitalia, you find that it's rather vague. It's not really true. There are variations within a species. There are different types of genitalia, and sometimes some species have different genitalia, and you have to agree that they're the same species. And some species have identical genitalia, and they're also the same species. So it's not really hard and fast. Then recently they came up with the thing of DNA. And they said, now we're going to make DNA maps of the different species, and there we will know for sure who's who. Unfortunately, the DNA thing depends on a sequence from a particular part of the genome of a species. And on comparing that, you are supposed to be able to tell whether two things are a species or not. In truth, how it works is that they compare these strands of DNA, and if there is a more than 3% difference, they assume that it is a different species. If it is not, they assume that it is the same species. Now, where the 3% difference rule came from, it came from nowhere. There is no basis for this. So in I think gorillas and humans, there's a very little bit of difference, and we are different species. And in some island populations of moths, you have more than 3% difference, but it's very clear that they belong to the same species. The point I'm trying to make is that the three methods we have of distinguishing species, which are the foundation blocks of biology, are all unreliable. There's nothing hard and fast about defining a species. So current thought, you get to hear, oh, the DNA of those was different. The DNA is checked, it's confirmed it's a different species. No, the DNA doesn't confirm anything. Unless the difference is vast, then it might confirm something. But otherwise, it is only an opinion of the person interpreting that data. The physical appearance of the creatures, the genitalia, the DNA, these are all indicators, but they need interpretation. It's data. But everything depends on how that data is interpreted. And it still depends on the opinion of taxonomists, whether something is a different species or not. The actual rule of thumb would be to actually breed those things and see what happens. But first of all, we don't have the time. And then second of all, you have rather silly definitions popping up, rather silly definitions of species popping up. Some years ago, there was what they called a phenological concept of species. Now, what they did is that you might know that we have a single antelope in India, the black buck. But in Africa, there are very many. There are dozens of antelope species there. Now, those antelope species occur in different parts of Africa. And in some cases, the different populations have different horns. So this group of American researchers, they came along and said, well, we are going to state that each of these different horned species are different species, so that conservationists will actually protect them. Because at the moment, the conservation plans are species-based. And for example, if you have Thompson's gazelle in Kenya, which has straight horns, and they say, okay, this is in a protected area, but the Thompson's gazelle in South Africa might have spiral horns, and there's no protection for that. So they said, we'll make the South African Thompson's gazelle a different species and the Kenyan Thompson's gazelle a different species, and then both of them will get conservation attention. So their thing was that if a species can occur in different geographical areas, if there's no connection between them, they should be considered different species, even if they can mate and produce fertile offspring. That's the phenological concept of species. So there was a boom in species descriptions, for example, Himalayan and South Indian butterflies. They're isolated populations in the hills of South India, in the Nilgiris and the Palnis. There are many Himalayan plants, birds, mammals, reptiles, fish, butterflies, moths. So obviously these are all phenological species because they're not connected to the Himalayan population. And even if they can mate and produce fertile offspring, they are separate by 1,000 kilometers, 2,000 kilometers. So they are different species. The trouble arose when this concept was applied to humans. Because if the phenological concept of species is applied to humans, then every geographical variation of humans become different species. So we would have every tribe of humans a different species. That is completely unacceptable. So the phenological concept of species quietly slunk out. So you have these what did I do? I pressed a button. Can you help me with this? What did I do? Where do you cancel?
You might have stopped video, no?
Hello?
You might have stopped video, sir.
Yes, Havik. Hello, sir.
Sitter, sir.
Yes.
You, yeah. You
Yes, sir,
are,
we can hear you.
yeah.
For okay. So, well, basically, taxonomy is where we are trying to find out to distinguish the different species on the basis of the little bit of data that we can generate from them, whether it's DNA, whether it's the genitalia, whether it's the physical appearance, whether it is the habits. In birds, now they're recording the songs of the birds, and a thrush was recently distinguished because it occurs at a higher elevation and has a different sound, different song from the lower elevation thrush. You might say that these are dialects. In humans, you would say, okay, he speaks Pahari and he speaks Desi Bhasha, and they're the same species. But well, these it has been described as a different species on the basis of the different song. So taxonomy, species, we have to realize that when we're studying the biological world, the only thing that is constant and doesn't change in biology is the process of change. Because bio means life. Life means that you are active and you are adjusting to the challenges of your environment to survive. And that adjustment is a continuous process. So when we have a genome, which we want to compare with other genomes, we have to realize that even the genome keeps changing. You can say that we are four-dimensional. Life is four-dimensional. We have our three-dimensional world around us, and the fourth dimension is time. So we have to take time into consideration when we are talking about classification and anything, in fact, in biology, because it is all about change. Biology is the process of change. Otherwise, a rock doesn't change over long. Even a rock changes, but it changes slowly. And in biology, things change rapidly. So what is the today's wonder fix cure might be discredited tomorrow. 100 years ago, genitalia studies were the wonder fix cure. Today, they have been practically discredited. Now we've moved on to the DNA. Now, DNA, 100 years later, there'll be something else come up, and then we'll find out that, maybe DNA wasn't such a good idea after all. And all the new cryptic species that have been distinguished in this period might turn out to be the same thing after all. So one has to go at it rather carefully. Nothing is clear-cut in this field, and everything keeps changing. Right? You have to have your concepts clear. You have to understand what a species is, what a subspecies is, and what we are looking for, and then how we can modify them to suit what is of advantage to us. And whether it is controlling pests, or whether it is figuring out new viruses, or whether it is whatever. So we have to go at it carefully. Please don't accept anything at face value until you have examined it yourself. That is my experience, and which I would very happily recommend to you. Right? And I think is an hour over? Is it 12 o'clock? How much time do I have? Oh, it's only 11:36. I'm sorry. I don't usually talk so much. So, having said all this, we can talk about butterflies. We can put the volume down a bit, the music. In dogs, there's a very old, the different breeds of dogs. You have the Chihuahua, you have the Great Dane, and you look at them and they look completely different. So but they're the same species. Good. In butterflies, we have the same challenge. In other insects, we have seasonal variation, geographical variation. Many people, many students ask me about a subspecies. What is a subspecies? So we've just talked about a species where it is based on the ability to reproduce and produce fertile offspring. Subspecies would be geographical variations. In humans, we would say that Africans, Europeans, East Asians are subspecies. So we look different. If you put us together, we look different, but we can produce fertile offspring. So we are one species. So in many cases, we have butterflies that are found in South India or in Australia, and they look rather different from the main populations in the Himalaya or temperate Asia. So they are subspecies, right? They can continue to produce fertile offspring. And as our principal sir told us a little while ago about, and Neha madam, that I work on bioindicators, right? About it's not enough to know what a species is or give it a name. That is not where it ends. The process of giving a name, the namkaran, is the first sanskar that a child has, and after that, other things happen. So in the case of insects, or in the case of any life form, we can use the presence or absence of that life form to understand what is present or absent in the environment. We all need certain basic things. Now, for humans, we need 2 liters of water a day. So if we are in a desert and we see a family of 4 people living there, we assume that there is 8 liters of water available daily over there, right? So in the same way, we can use, if you see a lion needs to eat 10 kilos of meat every 2 weeks. So there is 10 kilos of meat every 2 weeks available for that lion. And for insects, they require certain food plants for the early stages to feed on, the caterpillars or the nymphs. They require certain humidity and temperature levels, because insects are cold-blooded. So they require certain temperature levels and humidity levels to survive. If it gets too hot, or if it gets too cold or too dry or too wet, they can't survive. And once we understand these parameters, we can predict whether an insect can occur somewhere or not. How should we say it? These are the basics: temperature, humidity, food plant. But there are very many other finer points where you might have the food plant and the temperature and the humidity, but the insect is not present. So it is in understanding these finer points that we can get a better handle on what is going on about the presence or absence of bioindicators. And we can understand the importance of particular small features of the environment. Now, I know this sounds like Greek to you, but I'll give you an example. We have a butterfly called a golden birdwing in the hills. So you have the southern birdwing in South India, which is the largest. It used to be the largest butterfly in India, but now the golden birdwing is the largest. Anyway, these large butterflies, they require to eat a lot in the caterpillar stage. Now, they all feed on a climber called Aristolochia, which is poisonous. Now, in the hills over here in the Himalaya, Aristolochia climbers grow all over the place. So you have them on hillsides, you have them in valleys, you have them near streams, and you have them on dry hillside forests. The difference is that when it's growing on a hillside, it grows about 1 foot or 2 feet high. It has about 20 leaves. and no flowers. And if it is growing next to a perennial stream, the plant grows about 20 feet high, and it has very, very big leaves, and it has flowers, and it has seeds. Now, the seeds of the Aristolochia look a little bit like a Loki or a Karela, or actually very much like a torei, a ridge gourd. And they're about 6 inches long, right? About that long. And they contain the seeds. Now, these seeds are seed pods only grow on the plants where there's enough water. Now, there are other butterflies that feed on the Aristolochia, but they are not found. The other butterflies feed on the Aristolochia. They're found everywhere. But the birdwing butterflies, birdwing butterflies are only found in certain forests. So one would expect them to be found everywhere, but no, that's not the way it works. And the reason is that the birdwing caterpillar needs to feed on the seed pod of the plant. Yeah. So if it doesn't get the nutrition from the seed pod, it doesn't grow into a big birdwing. I fed a birdwing caterpillar on leaves, and the chap came out a small fellow because he didn't get the nutrition he required. So to grow properly, they need the nutrition from the seed pod. Now the seed pod will only grow where the food plant is growing near water. Therefore, the birdwing butterfly is an indicator of perennial water. And if the population of birdwing butterflies in an area begins to drop, then it means that the perennial water has dried up. Those Aristolochia plants are not flowering, and they are not producing fruit, which the birdwing cannot eat then. Right? So the birdwing then becomes an indicator of perennial water. This is how we distinguish. Bioindicators. Yeah? And the small things like perennial water, the climbers, all of these add to a mosaic of what a forest ecosystem is. And if you consider that in India, we do not have a definition for the health of a forest. Nowhere in the world do we have a definition for what a healthy forest is. So it becomes important to understand that things like perennial water, climbers, certain flowers, they all form a mosaic which contributes to a healthy forest. Now, again, what is a healthy forest? How do I say that a particular butterfly makes it healthy when the same butterfly might reach such proportions that it becomes what is called a pest and does damage to the system? So a healthy system would be a system, a forest or a community of plants which is in synchronization with the local climate. So in the desert, you will have plants which survive on very low water budgets, and you will have plants that can lie dormant for 20 years, and the moment it rains, they pop up and flower and die down again. Right? So that is a healthy ecosystem in a desert. In a tropical rainforest, you might have very big trees and a lot of canopy, very dense canopy, etc., but very impoverished soil. So an impoverished soil is a sign of a healthy tropical rainforest, because all the nutrients are in circulation in the trees and in the organisms that live in them. So we don't have a fixed definition of what a healthy forest is. But what we do know is that if an ecosystem is adjusted to the water supply to it and the nutrients available to it, then that is stable over the long term, which is what we want. Now, how do humans benefit from a healthy ecosystem? For India, it's very simple, and that is that we have many rivers in our country, and our rivers begin and end within our country. All the peninsular Indian rivers, the Krishna, Kaveri, the Godavari, Mahanadi, they all originate in India and in India. So we control the same with the Ganga. We only have one tributary coming from Mansarovar. And the only river that actually comes from outside is the Brahmaputra. All these rivers originate in rain-fed watersheds. If we can control the forests and the headwaters, we can control the water budgets of the rivers, and we can make sure that
दूसरी मोहब्बत अक्सर उसी शख्स से हो जाती है जिसके सामने हम पहली मोहब्बत का रोना रोते
Yes, if we can control the headwaters, the water budget of the river, then we can ensure a stable water supply for our subcontinent. So that is where butterflies fit into the benefit of our nation. Now, how do you work that out? We all know a wash basin, right? Now, if you open the tap in the wash basin, the water flows off. That is what is happening nowadays. But if you put a towel in the wash basin, then it soaks up the water, and it soaks up the water until it is saturated, and then the excess water begins to go down the drain. And even if you shut off the tap, whatever is in the towel keeps dripping down the drain until the towel is dry. This is Bittu Saigal's example, which is a very valid example. And forests are towels in the wash basin. But what is a good towel for which wash basin, right? That is what we can be told by the community of creatures that plant community supports. And in the case of birds, you have birds that can fly 50 kilometers a day to get water or food, and they can fly over long distances to roost in a completely different place, eat in a completely different place, and drink water in a completely different place. So birds are good over large landscapes, but insects are local. Many butterflies will remain within a 5 square kilometer patch. And if we can assess what the population should be and their presence and absence, we will know all about the perennial water in that area and the vines and the this and the that, which gives us an insight into how the forest's health is in that area, which is what is the state of the towel. Over most of India, we get 80% of our precipitation between June and September. So in 3 months, we get 12 months worth of precipitation. We have to learn how to stop that precipitation and let it out on a trickle, and then our rivers will keep flowing healthy. At the moment, what's happening, 80% of the precipitation falls. It rushes down into the streams and rivers. We have a flood in the plains, and then we have a drought later on. We have major water scarcity in the summer months because the water that should have percolated into the soil and be held in forests has already reached the sea. So we have to rejuvenate the forests in the hills. But what forest? What is a forest, right? So we need forests which are supporting a community of creatures which are indigenous to the area. Not plantations, not monoculture plantations. So, would my co-hosts come online, please? I think I've said what I've had to say, and I've given a bit of my opinion on what the whole picture is. And if there are any questions to be taken, I would be happy to take any questions from the students. Oh my God, I've been, hello.
If any questions from participant, please ask.
What's the time?
Biggest butterfly in the world.
Hello.
Hello, Peter sir, can you?
Yes, hello.
Yeah, anybody wants to ask the question? The session is open to interact with our speaker.
The biggest butterfly in the world, and what is its lifespan?
Sorry, Vinod. We can have your question again. We were not able to hear your question. So please.
The biggest butterfly in the world, and what is its lifespan?
Oh, okay. The biggest butterfly in the world, and what is its lifespan? Well, that's Queen Alexandra's birdwing, which is found in Papua New Guinea. lifespan of butterflies is usually around 2 weeks. That is because the butterfly is the adult stage of a long process. The main lifespan is spent in the caterpillar stage eating, usually 2 months or so. And the adult stage is only for reproduction and spreading the species. So that's usually 10 days to 2 weeks. Right?
Okay, sir.
Thank you, Vinod. Anybody else? Okay.
Thank you, sir. Thank you very much, sir, for your
Hello.
valuable information.
Thank you.
Hello.
It will definitely help us in future.
It will be a
Now I
pleasure.
request Miss Mayuri Thakkar ma'am to propose vote of thanks. Please,
Good
ma'am.
morning, sir. I would like to express our sincere thanks to Peter, sir, for giving an excellent coverage in current trends in taxonomy, and especially talking on the topic of butterflies. Many people are fascinated. Zoologists are not zoologists, but many people around are fascinated with butterflies. So by choosing this topic, I guess many have known more much about the butterflies, as if they play a role in indicators. And as earlier you said regarding mimicry, many of the students relate to it. And I hope that the students and other participants would have really gained a precise knowledge with reference to taxonomy. Especially, I would like to bring to your notice, sir, that our MSc students are doing their specialization in entomology. So it would be a great helpful to them with this aspect of taxonomy. We are all inspired by your illuminating words. It has been a great pleasure. A big thank you to Peter sir for his effort. Thank you, sir.
Thank you.
Thank you, sir.
Thank you. Goodbye. Pleasure.
Bye. Now I will introduce next speaker for session second, Dr. Meghnad Doshi sir. It's my privilege to introduce next speaker for session second, Dr. Meghnad Doshi sir. Currently, he is working as Associate Professor at Department of Stem Cells and Regenerative Medicine, D.Y. Patil Education Society, Kolhapur, and also owns STEM Plus Biotech Private Limited company in Sangli as Managing Director. He did his PhD from Shivaji University, Kolhapur. He completed his postdoctoral and diploma and postdoctoral research in Department of Transplantation Surgery, Karolinska Institute and Gutenberg University, Stockholm, Sweden. He has published 33 research papers in various national and international journals with 321 citations on his credit. He delivered about 26 invited talks in various colleges, universities, national and international conferences and workshops. 5 PhD students awarded under his able guidance. Also, he has 4 patents on his credit. He has written 4 chapters in different books. He is a reviewer for Journal of Stem Cell Therapy and Transplantation. Now, I request to Doshi sir to share his views with us. Please, sir.
Extremely sorry. I need 5 minutes in order to start my PPT. I had some internet issues, so it will be a little bit delay. Just give me 5 minutes.
Meghnath sir, we are not able to hear you. Sir, we are not.
Give me 1 minute.
Yes, sir.
Yeah. Am I audible? Hello?
Yes, sir.
Sir, have you disabled the annotation over there?
Annotation.
You are going to present the screen, na? So
Yes. Just
while you present the screen,
I am having a lot of problems with this PPT. So what I'll do is I'll start for 5 minutes and immediately I'll start PPT. Maybe this is
yes, sir.
the share screening, right? Host disable the partnership. You have disabled the screen sharing.
इथं नाही दिसत, तिथं दिसते. सुरू कर म्हणून कोणतरी. हे. ठीक आहे. ओके. Yes, sir, you can start.
Yeah, okay. Okay. So what I'll do is I'll just start a little bit about the introduction because right now I'm downloading the PowerPoint, so maybe it will take 5 minutes for me to start on the PowerPoint. First of all, I would like to thank organizing committee for inviting me for this webinar. The first lecture was very interesting, and of course, though I'm a zoologist, I'm a little bit away from basic kind of research. So basically, I work majority on cell biology, but still, being a zoologist, I still have an interest for taxonomy. Thank you very much for this very nice introduction for the about the taxonomy. Today's talk, I'll be presenting on a little bit about the introduction to stem cells. What are stem cells? Why they are important in human diseases, and a little bit about the recent advancement that are tissue engineering and 3D printing. See, I think majority of the students are from zoology background, so maybe I think I should little bit introduce how the basic subjects are. The basic subject actually help you to build over your career. So I think I should spend some time for all of you because that's very important, because you are just going to be out from your graduation, and majority of you people are looking for the change in career. I strongly recommend that whenever people are pursuing, the basic degree will be very useful, even though it might be in zoology or botany or chemistry or say physics. Basic degree has its own potential because you are equipped with many things at the same time. For example, when I graduated from zoology, I took physics. chemistry. These are the things which equip you whenever anyone wants to go for a research in next way. So that is very important. Of course, I did my graduation with zoology, then masters in zoology and cell biology, and then completed my PhD in cell
Excuse me,
biology.
sir.
Yeah.
Sir, can you please increase your volume over there? Your voice is
Now it's on high actually.
highest. Keep it highest.
You could hear, right?
Yeah.
Okay. So this the basic point here is if you are you come from the basic science, you chances of you in getting to diversity of fields is quite a bit. So after completing PhD, I entered into stem cell field and then uh, for tissue engineering, 3D printing. So I still think that considering a basic degree in any subject has a huge potential. Of course, there are enormous research in biotechnology is going on, and I think this kind of newer technology will definitely help you to start up your career. I'm also associated with DY Patil University. I'm working there as an associate professor, and I'm heading. I'm also. I have also a biotechnology company which is situated in Sangli. So whenever, if you want to visit my company, you are most welcome. You could also check my information on website. Is on which is STEM plus cryopreservation and STEM plus biotech, which is available on the internet. So with this, I think I should begin my talk on advancement of stem cells and little bit about the application part of stem cells, that is tissue engineering and 3D printing. Now, whenever people are talking about stem cells, they have glorified it in a very broad sense. But as a basic scientist, I still consider that it has limitations, and the more and more you do the research, more and more things you will start to understand. So, with this, we should start our lecture. Okay, now share the screen.
So please, if you have a headphone, can you use it so that your voice will be more clear to all of us?
I need to check it. Wait a minute. Now everyone could see my. Wow. Just a minute. Let me put a manual one.
Yes, sir, we can see your screen.
So I'm audible, right?
Yes, sir.
Good. Okay. Thank you. This is good. Okay. Thank you. So let's start something regarding the stem cell research. Why stem cell research is important? Important. First thing is, when you are doing stem cell research, it's very important that you are actually studying the developmental biology. That means how humans are formed, how during the developmental stages, how the organs develop. So all those things are important, and one you could study it by checking the characteristics of stem cells. And most important thing is, when stem cells are used, they will be able to replace the diseased or damaged tissue, which actually in a normal situation where after the injury, these diseased tissue or organ cannot replace. So once you introduce those stem cells into damaged tissue, stem cells able to replace the tissues and this organ can be rejuvenated. There is also a newer trend in pharmacology where different drugs or chemicals that can be tested on stem cells, so that how the stem cells function can
Sir.
be. Yes.
तुमचा आवाज अजून कमी येतोय, जरा माईक जरा जवळ पकडला तरी चालेल, थोडा कमी येतोय आवाज. नाही, आवाजच येत नाही आता.
Hello, Ata?
या.
I think I'll go a little bit close. I think.
Yes, sir.
So more, then another part is about our genetic machinery, because during disease progression or specific population has specific disease, so we will be able to study the genetic machinery behind the disease with when you study stem cells. So all those points are very important when it comes to study of stem cells. So there are specific characteristics of stem cells. That's why they make these. Every cell is has the same characteristics. But how you are going to identify normal cell and stem cell? Wow. Normal cell and stem cell. First thing, whenever a stem cell divide, it gives rise to another stem cell, and at the same time, specialized cell, which can be hepatocyte, muscle cell, or nerve cell. So whenever a stem cell divide, they renew cells. That is, once the stem cell divide, another stem cell is being.
Sir, are you changing your slide?
Sorry.
Have you changed your slide?
No.
Okay. Otherwise, keep slideshow करा.
It's a slideshow.
Slideshow नाहीये, तुमचा PowerPoint view दिसतोय.
होय, पण slideshow सुरू आहे आता.
From current slide म्हणला ना? Okay.
I think you should ask your IT person, because right now I'm
You're on the title. Your title screen, na? Recent advance.
Sorry.
You're on the first slide only, na?
No, I'm on the third slide.
So we are not able to see that slide, sir. Sir, please press the F5 button. F5.
Yes.
Sir. इथे त्यांनी इनेबल एडिटिंग मध्ये नाही, हे काय दिसत नाही. हो ना, पण मी दिसत नाही. Yes, sir, we can see now. Yes, sir.
Now, right now, what I did was I just exited the full screen.
Yeah, that's. But now we are able to see now this stem cell have unique characteristics. Yeah.
Now?
Yes, sir.
You could see full slide, right?
Yeah.
So I'm getting these messages constantly. Can you mute it?
Message stop करू शकत नाही.
I think you have made me admin participant. I'm just getting pop-up messages. Okay, let's come back to the point. So when stem cell divide, it will renew itself. That means one stem cell divide, another stem cell is formed. At the same time, there is a specialized cell that is being formed after the division. Okay. So there are three important functions of stem cell. First thing is the plasticity, that is mean potential to change into other cell types like nerve cells or hepatocytes. Homing, that is able to travel at the site of injury so that the regeneration can be triggered. And engraftment, that is able to unite at the site of transplantation. That means whenever you transplant stem cell in the liver or kidney, the stem cell able to integrate with the native tissue. That is, once you transplant it in liver, it should be able to unite with liver. So this is a stem cell potency classification. That is, potency is something.
Excuse me, sir.
Yeah.
तुमची स्लाइड पुढे मूव्ह होत नाहीये, तर F5 बटन दाबा.
F5 बटन दाबले मॅडम.
ही स्लाइड पुढे मूव्ह होत नाहीये.
So let's begin like this only.
Yeah, sir. Keep it safe.
I think that's better. You have a lot of problems here.
Okay.
So you have stem cell potency classification. Potency is the functionality of any stem cell, and potency of stem cells get reduced with age. That means with age, cells become less and less potent. So at the higher side, totipotency is known. That totipotency means single cell able to generate complete embryo. That's in the say if you have a single cell fertilized cell, and if you want to if you isolate that cell, this cell able to form a complete embryo. Now with age, this totipotency is lost and cells become pluripotent. Pluripotent cells can be also found in inner mass of blastocyst, embryonic stem cells, or induced pluripotent stem cells. So these are the pluripotent stem cells. So pluripotency is classified as stem cells able to differentiate into 3 germ lines. I think being a zoologist, you all know that germ lines, because we have studied different developmental biology very much. So we all know theory of 3 germ lines. So 3 germ lines, the pluripotent cells able to differentiate between the 3 germ lines. So with age, pluripotency get lost and then cells become multipotent. So multipotent cells are cord blood stem cells, bone marrow stem cells. So these stem cells able to differentiate between the single germ line. So with age, that is also been lost and cells become unipotent. That is, unipotent means able to differentiate into specific cells, or something like special cells in the organ, where these cells able to differentiate between the cells of the same organ. So this is how you classify the stem cells. This also classified in animation forms. So these stem cell sources and stem cell potency is divided into 3 different things. First thing is the embryonic thing. The cells are totipotent cells, but they are age dependent. If you isolate the cells in 3 days, 3 days after fertilizing, cells are totipotent. That means even though you isolate single cell, these cells able to differentiate into complete embryo. So this is the characteristic of totipotency. So this has the highest potency. If you could see the below triangles, you have progressive loss of potency and developmental stages. So the cells become more and more specialized with development, and the totipotent cells or embryonic stem cells have the highest potency. So during the aging process, the potency get lost. So another class is fetal stem cells, where you could find pluripotent stem cells. So fetal stem cells can be isolated from fetuses, and these fetuses are maybe legally aborted fetuses or embryonic cells. So you could find cells from the day 5 to day 14. So cells are pluripotent cells. They have able to differentiate between the 3 germ lines. So this is the characteristic of fetal stem cells. Now these cells have a lesser potency, but at the same time, these cells have less developmental characteristics. So the cells are not more specialized. Now the next class of cells is adult stem cells, where you could find multipotent cells, oligopotent cells, unipotent cells. Now this age of cells can be from 9 months to 100 years. So at the same time, cord blood is considered to be a less age cells, because the age of the cord blood is 9 months. So the cord blood has the 9 months age, so that is the highest in the potency ratio. So in adult, whenever we are talking about the adult cells, cord blood has the highest potential in terms of stem cell potency. So then comes bone marrow, then there are unipotent stem cells. So there are different kind of cells what one could find it in adult life. So what makes stem cells so valuable? So there are different things, applications that makes them very valuable. First thing is cell therapy. So if you have a disease patient and there are no current treatment scenario, stem cells is the ideal candidate for treating these patients, and you have wonderful results using stem cells for different chronic diseases. Of course, it has a huge challenges when it comes to research. These cells are of interest to geneticists, molecular biologists. Of course, you could actually understand in vitro how the cells function, how the disease progression happens, what are the major factors that can be influenced by stem cells. And of course, as I discussed, new drugs that can be easily treated, easily being discussed in petri dish using stem cells. Of course, there are a variety of different diseases are being treated with stem cells, and more knowledge is compiling throughout the research worldwide. But still, I would like to comment that no one stem cell type fits for all applications. Research must continue using all types of stem cells. So there are still limitations on using stem cells, and there is a need of more research in order to understand what are the specifications of stem cells required for applications. So this is very important when it comes to defining stem cells, because as a zoologist, we should understand how stem cells can be how you are going to identify stem cells. So there are, of course, there are some gold standard methods where one able to identify stem cells, and they can be identified by the expressive marker on the cell surface called cluster of differentiation, or in brief it's called CD markers, and they are considered to be a gold marker because every cell has its own cluster of differentiation that is expressed on the cell membrane. And with the use of fluorescence microscopy, you will be able to identify between the cells, and you could actually identify how different stem cells are been expressing what different kind of CD markers. Now, in all, when you are culturing stem cells, you will be able to identify the same. You will be able to see the stem cells. You will not be able to identify between the stem cells and adult cells. For example, in this first picture, this is hepatocyte, adult hepatocyte cell.
Hello, sir. Excuse me, sir.
Second slide has a mesenchymal stem cells, and these are also adult cells. Now, if you are looking under phase contrast,
Excuse me,
hello,
sir.
हॅलो
हॅलो, हा सर, ते अनोटेशन बंद करायचं होतं. जे रेषा मारतात ना मुलं, तर ते बंद करायचं होतं अनोटेशन, प्लीज.
ते कोण मारा अनोटेशन बंद
तुमचे
करायचं?
हो, तुम्ही क्लिक केलं का, स्क्रीनला क्लिक केल्यानंतर वर जो बार येतो ना सर, तिथं राईट साईडला आहे ना, वर एक ऑप्शन आहे अनोटेशनचं, 3 डॉट आहेत, 3 डॉट. हा, डिसेबल युअर अनोटेशन. हा,
डिसेबल आहे बहुतेक.
रेषा स्क्रीनवर ज्या रेषा मारतायत ना मुलं, बाकीचे कुणी काय करतात माहिती का, गोंधळ करते, रेषा मारते.
एक मिनिट परत चेक करतो.
वर राईट साईडला 3 डॉट आहेत सर.
Okay, sir. Thank you.
Yeah. So whenever we are looking at cells in petri dish, it is really difficult for us to identify between stem cells and normal cells. So it's better that you identify it with using different CD cluster of differentiation markers. So in all, whenever you are going to study between the different stem cells, they generally classified into three lines, that is epithelial cells, mesenchymal cells, and endothelial cells. Epithelial cells are secreting cells like hepatocytes. Mesenchymal stem cells are stromal cells that they able to support the structure, and endothelial cells are the simple cells, something like blood vessels or any kind of margin of organs. So these are endothelial cells, and they have different cluster of differentiation markers, something like CD34 for epithelial cells, CD133, CD90. As well as for mesenchymal, there are different markers which are being put here. And of course, endothelial cells have CD34, VGFR2, Ti2. So these are few of the markers which are being seen on the cells.
बर.
So this is
पण
typical.
मग ते आपल्याला म्हणजे दररोजचं येऊन आता हे 6 दिवसाचे त्याने पिक केलं का आपण केलं?
Hello.
So 1 minute.
तुम्ही करता, पण मग येतो का तो बरोबर? हा मग ठीक.
So this is a flow cytometry characteristic of mesenchymal stem cell. Flow cytometry is a technique where you actually label the cells with antibodies, and these antibodies are linked with fluorochrome. And whenever you pass those cells through flow cytometer, single cells are being analyzed for expression of markers. Say for example, if you have a mixed population of stem cells and you label them with different antibodies, so in a single population you will be able to identify the population of different stem cells present in the say for example stems, bone marrow or cord blood. So this is also advanced kind of a technique which is being used in the medical field. There are also techniques like you could actually visualize expression of stem cells. For example, here we have characterized the fetal hepatocyte stem cells using immunocytochemistry. So these are the green fluorescent protein is expressing albumin. For second, here is the alpha fetoprotein, because these are the fetal stem cells. So they express fetal protein. This is also a receptor called CMET. So these cells express CMET receptor. These cells do not express CD271 because they are not epithelial cells. So at the same time, the characteristic of epithelial cells are being seen in the other expression like cytokeratin 8, cytokeratin 18, 19, and most importantly, the fetal is immunocytochemistry, and this is the flow cytometry. So flow cytometry, actually you will not be able to visualize it, but the expression of the stem cell is being converted into digital form, and this is being seen with the dots. Hello.
Yes, sir. We are able to hear you.
I'm hearing a lot of noises, background noises. Now I'm going to present you some of our recent work on kidney tissue engineering, because we work a lot on, my lab work on a lot on tissue engineering techniques, and we work with different organs for where we actually engineer everything in the lab. So the tissue engineering field actually has a huge potential, and when I was in Sweden, I was involved also in a clinical transplantation where you actually engineer an organ using patient stem cells and transplant it again back to the patient. So after that, when I started to form my own group, we have used this technique for developing different organs for transplantation. And here I'm going to present something regarding the kidney tissue engineering, because kidney has a huge potential when it comes to when it comes to tissue engineering application, because the chronic kidney diseases severity is very high in India, and unfortunately, transplantation is the only available treatment option for these patients. And availability of organ donor is very less in India, because majority of the time India has a living donor. That means your sister, mother, or your close family actually donate organs for you and a patient, and they get transplant. But considering the present situation where in the globalization our family structure is being disturbed, and many patients they are unable to get a suitable donor, and these patients die in a waiting list because they don't get a transplantable organ. So what we thought is, if we could engineer something for a patient specific, that means if we have good quality of stem cells from the patient, and we will be able to have a scaffold where these stem cells can be cultured, and you could actually engineer a transplantable organ. So we have taken one step for this, and we have checked how this tissue engineering technology can actually work, or how it could be actually developed in terms of clinical reality. So what we did was, first thing, it was the generation of tissue engineered scaffold, because scaffold is a material where cells can be seeded. That means you have stem cells, but you could able to culture it on this scaffold, and these cells actually able to differentiate into different organ specific lineages. So this is the goat kidney which we have, which is the starting material. We actually wash it with antibiotics so that the blood remnants will be removed, and slowly, with using detergent-based technique, actually we wash the entire kidney so that there are no cells available in the kidney. So during the perfusion reaction, what it comes, see, if you could see during after the 52 hours, the cellular content, that is goat cellular content, is been completely removed by the detergent based system, and you could actually check it using actually renal angiogram. This is the x-ray of kidney, where you could actually see the blood vessels present, and there are no other cells present in kidney. Now, this also can be visualized using scanning electron microscopy, and this scanning electron microscopy, you could see actually a preserved membrane, and the skeletal system of kidney is present. And this also can be checked using DNA content, because every cell has DNA. So if you are washing it, or you are removing the cellular content of that kidney, this is checking the DNA content will be the ideal marker. So one could actually check the DNA content in the kidney and determine whether you have removed goat cells or not. So what we did was, after 52 hours, all the goat cells are being removed from the kidney. So once you remove the goat cells from the kidney, so what we planned, we would like to check whether this generated scaffold, that is decellularized scaffold, is an ideal matrix for generation of kidney. That means, if we transplant this kind of a matrix in a rat, it should be able to facilitate the development of kidney. So that what we did, surgically, we have removed a part of kidney and placed the scaffold, a small piece of scaffold in a kidney capsule. What it happens is, if this is also called a xenotransplant, because we have taken a scaffold from goat and transplanted into rat. So this is also called a xenotransplant. So what we actually aim in this entire process is, actually you could engineer something like a human size, and able to generate customized organs only using scaffold. So what it happens is, if you implant that scaffold into kidney capsule, it actually stays at the site, and after 30 days, still you have a kidney capsule which is being easily, which is functionalized, and it has a lot of blood supply. And what we did was we have checked this scaffold to understand what are the cellular content of that kidney. So during the process, we actually check different MRI images in order to understand whether the transplanted kidney scaffold has a living kidney or not, because there is a possibility that even though you transplant xenogenic organ, you actually end up in rejection. So it is always better that you check if you have a functional kidney. So we could actually determine the functional kidney using MRI. So these are the different planes of MRI. So by this way you could actually check that there are the functional kidney present. So during the histology, you could see the eosin hematoxylin, Meisson's trichrome, AB staining, ripen blue staining. These are different staining in order to check the cellularity of the transplanted scaffold. So this is a control kidney where you have a entire structure. You could see the glomeruli, you could see proximal distal tubules. Now this has sorry. So this is the implanted scaffold. What we have did is we have magnified the implanted scaffolds, and actually you could see after the 30 days, the glomeruli start to appear. Of course, there are very few proximal and distal tubules, but majority of the time glomeruli has been already started to recellularize it. So this we have confirmed with different staining. For example, here AB stain, you could actually see a complete glomeruli here. uh, uh, TB staining also confirms that there is the cellularity of the scaffold has increased. That means the rat cells are infiltrated in the goat scaffold, and they actually started to recellularize, or they started to develop entirely new kidney, which is actually of goat origin. Then we also characterize it with different staining techniques. Here, for example, here immunohistochemistry, you could actually this blue staining is for nuclear content, that is stained using DAPI, that is a fluorescent dye present in marine algae, and which you could actually use it in fluorescent staining. And if you could see the green fluorescent that is actually your protein. So AQP1 is a protein which is exclusively present in the kidney epithelial cells. So expression of APQ1, presence of APQ1 actually gives you the idea that you actually are having a kidney specific cells. Of course, nephrin is also antigen present on nephrons. So expression of nephrin in this scaffold also gives an idea that there are kidney specific cells. So this is also alpha SMA is alpha smooth muscle actin, because every organ has a smooth muscle. So expression of SMA again denote the physiological functioning of this kidney. And VEGF is the endothelial marker, that is a blood vessel marker. And if you could see the expression of VEGF positive cells, that means there are blood vessels that are being formed in this scaffold. We have also confirmed everything, the presence of different cells using electron microscopy. If you could see, these are the glomerular cells which are present in the scaffolds. This is the entire glomeruli. This is endothelium, which has been seen. So there are also specialized kidney specific cell like podocytes that have been also found in electron microscopy photo. So there are Bowman's capsule is been present. So all these denotes that you could actually use this technology to develop a human-sized kidney using xenoscaffold. So this is how the preliminary data of tissue engineering work in my lab. Now we started to use the same kind of a technology in a bigger model. For example, a rabbit model, where instead of a small piece of kidney, we will be able to transplant the entire kidney. So the entire aim of our study is to generate human-sized kidney, which has been suitable for human transplant. So the basic idea behind this technology is if a patient has a kidney disease, and if you want to engineer something for this patient, you would actually get the xenoscaffold maybe from goat, and these goat cells are completely removed and make a opaque scaffold for human cells, and then you actually culture the patient specific cells onto that scaffold and make a patient specific kidney which will be able to transplant after the completion of engineering process. Now I will give you a little bit introduction about 3D bioprinting. This is a newer kind of a technology which we are using in my lab, and this is kind of a printer where you actually able to print 3D image with 3D printer. So in all what you have a 3D image. You actually send it to the 3D printer, and 3D printer will print a 3D image for you, and you have slice by slice printing of two-dimensional cross-section printed image. So 3D printing, it is easy to use, economical to once, can be operated in office, lab, homes. Actually, it is been equipped for students with skill of future effort, future technology. Of course, it is affordable in price when it comes to demonstration. It actually takes the digital 3D data and creates a solid 3D parts. It extensively used by designers, engineer, biologists, hobbyists for a concept development and a product design. So what we do is it's FDM technology, that is the filament technology, where uh, you have a material at this side that is in terms of filament, and then actually you liquefy with heating, and then after heating actually this nozzle start to pour or start to print this material into slices, and thus you get different kind of structures. Of course, 3D printing are been very interesting field in medical, because during the medical study you need to have a prototype for entire organs or in orthopedic situation, and use of this 3D printer can be an ideal candidate for making prototypes for study. So this is the first important thing. Now, these prototypes are also can be used for treatment, and now this is also a reality where 3D printed organs or tissues are already transplanted, or they have a clinical reality. So I would like to give you some of the interesting fact here. So during the medical process, they can be used in hearing aids or braces. They can be used for printing body parts, including ears, hips, even organs in except portions to fit to the patients. A first 3D printed clinical trial was done in February 2012, where surgeons successfully implanted entire titanium jaw made by 3D printing in an elderly woman. So this is how actually happens, that you have a titanium jaw and the entire half face, and we actually you could actually fit it as per patient need, so that this will look like same thing. This is also true. Just a minute. Yeah. Okay. So this is a typical example how the 3D printing can be applicable in human situation. For example, this as a patient had a massive face injury, and actually whatever you see here is wow, this movie is unable to run. Okay. So what happens is I will check. I'm really sorry, I cannot show this. So the entire part of his face is been lost during the accident. So what clinician did was they have printed this part on 3D printer, including eyes and eyebrows, and they fit it into the person so that it will be look like a normal person. So this is how the potential of 3D printer is. We also have developed a 3D printing technology in our lab for different purposes, something like for bone and bone applications. We are also working on skin, cartilage. So I thought it's better that I will show you something about bone printing. So now, whenever you are considering the bones, they are the rigid structure of body, and they actually protect the vital organs, and actually they give support to the body, and they also interact with muscles, tendon to generate body movement. So they have a central role to play. So bone injury due to trauma, fracture, degeneration, infection, abnormality resulting in long-term deformity such as bone shortening and compromised bone structure. So this is a major limitations when it comes to orthopedic surgeons, where in order to correct any kind of bone deformity, actually he has to cut it. And once you cut it, there is no replacement available till date in order to fix the situation. So what we did in our lab, we have developed a liquid bone technology, which is actually injectable in form. So it is when you are doing printing, it is actually liquid, and once you print it, it becomes solid. So solid like a rock. So this is how we developed here. I think this is also again.
सर तुमचा आवाज थोडासा कमी येतोय, जरा स्पीकरच्या जवळ राहा.
I think there are a lot of things here. So this is a 3D printer which is printing a bone tissue. So what it happens is, while it prints, actually it is a liquid form, and once the printing is been finished, so within a minute it becomes a solid structure which has been mechanically stable. So in all, electron microscopically you could actually see the bone structure, like it has a porous structure, a lot of cracks, and these are different calcareous material which has been evenly distributed throughout the bone structure. So these bone structure are being right now are being used in a different settings in order to check how these bone structure can be used in animals, or in a first phase, of course, animals. So maybe you have studied developmental biology like chick embryology, and we also use this tool to study the biocompatibility of the tissue engineered or 3D printed tissue. So what we do is we actually put a 3D printed scaffold onto cam area and check whether this 3D printed bone has biocompatibility or not. That means if it has a toxic effect, the embryo will die, and thus you could actually confirm that this is a toxic. So during 4 until 15 days, whenever you keep this bone, actually it is in a stable form. And we have also seen that during the later stage, we have seen it till 24 days, and actually the bone entirely get absorbed in that time. So this is also bioabsorbable material, so that it doesn't have any toxic effect. Of course, when you see this this bio ink, you could actually see that whenever you put this bone material on the cheek, actually it has a biocompatibility. That means cells started to come into the bone structure. For example, here you could actually see RBCs which are being infiltrated in this bone. So actually, what it happens is it has a porous capacity. That means different cells, they are able to engraft at the site of bone scaffold, and then able to actually recellularize with bone cells. Now, whenever we are talking about tissue engineering or 3D printing, the main zest of entire research is making a better scaffold which will actually integrate with the environment. So what we have, there are future research goals that that are very important in order to develop. So first is to develop more refined printers which can print smaller details, thus eliminating the need of a separate vascular system for organs, because you at any organ which is very complex, and you should be able to print it using different nozzles or different material at the same time, so that you could actually engineer the complex organ. Now, this is we need to check whether the 3D printed bone is strong enough for implantation. Now we are planning for animal experiment where we are going to replace the bone with boning and check whether how it is mechanically stable. Of course, longer lifespan, better condition of organs themselves, and it will be actually used in the medical field. So after studying the lifespan and the better conditioning of organs, we will be actually step forward for the clinical use. Of course, reduction of cost is very important when it comes to India, and working it more friendly or more economical, so that this technology can be used by clinicians or for the people. Thank you very much.
If any participant has questions, please ask. If any participant has questions, please ask.
Hello, Joshi sir. Just I want to first of all thank you for such a informative talk on stem cells and 3D printing. And this, I think, hope the students have also received something that is new and trending now in the science field. And about 3D printing, it's not known to all our students or most of the participants. And so you have shed the light on this trending technology. So thanks a lot. Just I want to ask one question, that during 3D printing, okay, so what kind of material, if you want to do 3D printing of a bone as I've shown, so what kind of material we have to provide to that, with that input given to that 3D printer?
Printing is material is it's called the bio ink, because
Hello, so your voice is very low. Can you go closer to the speaker?
The material used for 3D printing is called bio ink, and this
Bio
bio
ink
is separate for different organs. For example, you have a different bio ink for skin, you have a different bio ink for cartilage, you have a different bio ink for bone. So according to the printing organ, the bio ink changes, and thus use of different bio inks, actually you could print organs.
Okay, so for example, if you want to print heart, so we have to use different bio ink.
So you need a different bio ink which will facilitate the development of muscle.
Muscle.
So right now in my lab we have developed something for skin, we have developed for cartilage, and we are developed something for bone. See, there are a lot of limitations for 3D printing, because specialized organs like heart or liver, they have different functions, and presence of cells is also different. So that's what my last point was. We need to refine more onto specialized cells, so that you will have a better printed organ.
Okay, so from where we can get this bio ink?
You actually develop it.
We have to develop it. Okay.
It's the lab one right now.
In the lab?
Yes, it is our own technology.
Okay, sir. So apart from your lab, where the work is going on?
In India, there are 2-3 institutes in IIC, where they are working on 3D printing.
Okay. So do you provide any training regarding this 3D printing to any?
Not right now, but of course we are planning.
Okay, yeah, planning. So definitely our participants can make use of it.
Definitely.
Put all this kind of research methodology in your research projects, and try to invent some new things to the society for the benefit of society. So definitely, sir, thanks a lot.
Thank you.
There is one question from Sandeep Kumar chat box. Essay note: the pacemaker of heart can be made by the. Yeah, I think we have discussed it earlier now only. So do you want to answer him that whether essay note pacemaker of heart can be made by stem cells?
Wait, you have to again. I don't see any chat here. Chat, yes, wait. Okay. Okay. I don't see any questions. I don't see any questions.
It is in the chat box. One Sandeep Kumar has asked whether we can prepare essay note from the stem cells.
Whether we can. Can you repeat? Sorry, extremely sorry. Can you repeat the question?
Okay, he's asking whether essay note, the pacemaker of heart, can be prepared with the help of stem cell.
Yes. It can actually print it, but see, whenever we are talking about pacemaker, it has an entirely different application. Actually, it actually stimulates the cardiomyocyte. So you could actually print anything with printers, but of course, it will not be a bioprinter.
Okay, okay. Any more questions from the participants? Okay, we personally contact him for any purpose. We have his contact number.
Yeah, of course. Shall I type
Call
it?
him.
Shall I type it here?
Sorry, sir?
Shall I type for students?
Yes, sir. Yes, sir. You can type in the chat box. We'll personally contact you. Sir is providing his email ID, all the contact details in the chat box. And all the participants have to make use of this.
Yeah, both.
Yeah. Thank you, sir.
Thank you.
Thank you, sir, for your valuable guidance. Now I request Mr. UK Adam, Miss UK Adam ma'am to deliver vote of thanks and to conclude the session.
I am here for vote of thanks. It's an honor to offer a vote of thanks on the occasion of the national webinar meeting in our college. On behalf of my college and the entire management group here, I pay my gratitude to all the speakers for sharing their grateful words with us in today's meeting. The honorable and respected speakers, Mr. Peter Sir and Dr. Meghnad Joshi. I am thankful to all the participants for joining and attending the webinar. I take this occurrence to thank the entire committee, Honorable Mrs. Minatai Jagdane, Managing Council Member, Ray Shikshan Sanstha Satara, respected Dr. N.S. Gaikwad, Principal, RB Narayan Rao Borake College, Sri Rampur, our HOD, Mr. B.N. Kalunke sir, and all the members who extended their help in organizing the webinar. I must remark a proficient sense of gratefulness to all our guests for sharing with us their views and provoking address. All the participants of this meeting are all inspired by your highly sparkling words. I want to extend my generous thanks to the entire guests for their support and cooperation. It's not possible to thank everyone with words for such an appreciating involvement and the willingness they have expressed to finish the task. It's grateful to thank the members of the meeting for their valuable presence. With Principal Sir permission, I declare that the program is over. Thank you.
Thank you. All the participants are requested to fill the feedback link. Okay. Unless and until you don't fill the feedback link, you won't get your certificates. So fill it now. Okay. The link has been sent on the chat box. So thank you all the participants for attending this national webinar. So on behalf of Rare Shikshan Samstha's RBNB College, I thank you once again and see you soon next. Thank you, Joshi sir.
Very